Welcome to today's live Q&A where we're going to talk about how to prioritize your experiments. I'm Alexandra Schnoes. I'm an Associate Director of Career and Professional Development at iBiology. I'm a co-producer of the online courses on the iBiology Courses platform, including Let's Experiment, a guide for scientists working at the bench, and Planning Your Scientific Journey. And Let's Experiment is sort of the inspiration for this Q&A today. Behind the scenes, I'm also joined by my co-director, Shannon, and Noah Green, who's our course manager, who are going to help me manage your guys' questions and the chat function. So definitely, please don't stop yourself from putting in any questions in the Q&A or the chat. So I'm going to introduce our guests, but I will talk a little bit about our housekeeping. So if you look at the bottom of your webinar window, if you want to ask questions of the speaker, please click on the little thing that says Q&A, and it'll open up a chat box where you can enter questions, and you can actually also upvote questions that you think are great, and we'll be monitoring that and asking those questions as they go. You'll also notice that there's a chat box down there, and that's a place to put in things about technical issues or some other issue that's coming up with the webinar, not questions directly for the speaker. And we are live streaming this actually right now on our YouTube channel, so feel free to share with your colleagues who may not have been able to register. And we should have the links for the YouTube channel already in the chat box. If you don't see it, could someone write in the chat box and let us know if you're not seeing it? Okay, those are our basic housekeeping. Again, any questions, make sure to ask in chat. Let's get to the fun part, which is introducing Prachee Avasthi, who is our guest speaker and is an assistant professor of anatomy and cell biology at the University of Kansas Medical Center. So using a unicellular green algae as a model system, Prachee has a NIH-funded laboratory, which I think we're going to post in her lab website. And in her lab, she uses chemical biology, biochemistry, genetics, and quantitative live cell imaging to uncover novel mechanisms regulating the assembly of the ubiquitous cellular antenna. So little cute green algae with cute little antennae. Also known as the cilium, which Prachee can talk a lot more about, but that's not why we have her here, because we want her to talk about sort of career and professional development issues, which are things that she's also incredibly passionate about. She's a strong advocate for improved publication practices and serves on the board of directors for ASAP Bio and eLife. She founded the online peer mentoring community for junior faculty, the new PI Slack, and advocates for early career scientists on the steering committee of Rescuing Biomedical Research. Prachee received her PhD in neuroscience in 2009 from the lab of Wolfgang Baer at the University of Utah, and she then completed her postdoctoral studies with Wallace Marshall at UCSF, the University of California, San Francisco, where iBiology is actually co-located. So some of you will have already noticed that Prachee is a speaker featured in our course, Let's Experiment, a guide for scientists working at the bench. She is an amazing font of great information, and which is why we asked her here to talk about how to choose your next experiment. So thank you so much, Prachee, for joining us. We're really excited to have you. Is there anything else you'd like to add?
No, you covered everything. So I think you made my job easy on that part, which that's always the hard part for some reason, I think, compared to talking about the actual topic. But thanks for the kind introduction.
Our pleasure. We are just so excited to have you. So thank you so much. And now onto the topic. So as all of us who have been at the bench or the dry computational lab know there can often be a number of ways to experimentally address a question, but it is so hard to know which experiment you need to do next or next week or in the next month. So how do you structure and process that? Many scientists struggle with this question. I think probably all scientists struggle with this question. And not thinking this through through methodically, there we go, that's the word, could lead to a lot of wasted time, wasted resources, and that's particularly painful when you're a grad student or a postdoc. So to help students navigate this question, you actually developed a decision tree flow chart that we fell in love with and that you shared on your blog. And for the audience, we're going to share the link actually to the blog in the chat box if it hasn't been already. And we actually took this flow chart and put it inside our Let's Experiment course, along with a discussion on how to prioritize experiments. And unsurprisingly, we got a ton of response around this flow chart, a lot of questions, a lot of really thoughtful responses. So we wanted to have this Q&A to address some of those questions and open it up to the general public to sort of bring their own questions and bring them to you. So I'm going to start off with a couple of questions, but then we're going to open it up to questions from the audience. And just a reminder, type your questions in the Q&A box. And don't forget to upvote questions that you like. So Prachee, my first question for you is, what was the inspiration for developing the flowchart and prioritizing experiments, and why did you go to the trouble of actually writing it up and sharing it on your blog?
Yeah, so this is great, and I think you sort of really set me up really nicely for this question, just in talking about the fact that everybody has to do this, and it comes up all the time when you have a million different experiments you could be doing, and how do you decide what's the best use of your time? So it's not that we could really do every single thing that we want to do, so there has to be some way to decide, right? We only each have two hands, and it's a challenge, right? And so I think the thing that really inspired me to do this was just conversations with my own lab. And, you know, like everyone, I think that you probably are very familiar with this experience where you have conversations with your advisor and, you know, there are a lot of good ideas being thrown around, right? There's sort of, well, you know, everyone's excited about all these different experiments. And this happens to me all the time where I'm really excited about an experiment. And then maybe I'll read a paper and I'll get excited about some other experiment, or I'll hear a talk and then I'll get excited about another experiment. And all the time I'm sort of throwing these ideas out there. And I can see how that can be really challenging for, you know, for students to say, okay, well, this is really great, and now here's another thing that's really great, and here's yet another thing. Well, which one am I supposed to be doing, right? And so I think, you know, I used to have a lot of those conversations with the people in my lab, and rightfully so. There was like, well, which one matters? What's more important? Why do we, and, you know, and so. But almost all of the time, I think and I wrote this in the blog post too, that almost all of the time I would have a really strong opinion about it. That, well, of course we should do this one, or of course we should do that one, and this would be, you know, that's not to say that it's easy to decide necessarily, but I found myself sort of naturally, you know, given the information that we have and knowing sort of the situation with the resources and knowing the situation about timing, and a lot of these different factors come in. So that, you know, I sort of wrote the flowchart in a way that sort of talks about it almost purely from the experimental standpoint, but in many cases, maybe an advisor will have other factors that they're considering, right? You know, and it may involve, you know, like you said, resources and, you know, the career stage of the person and like experience of what types of experiments and what type of expertise they have and things like that. So that can vary a lot. And so I can see definitely there's scenarios in which even outside this framework, there's some decision making to be made. But it was very clear that, you know, it shouldn't be intuitive. It's not intuitive how to decide these things. And then I started thinking, well, why is it that I have a very strong opinion, and what is the basis of this opinion? So I thought, well, let me just sit down and write out, you know, what in my mind am I thinking when I say this is more important than that? And so I tried to sort of make this decision tree and say, well, these are the questions I'm asking myself, when I'm thinking about it. So it happens a little bit more seamlessly in my head. But if I were to deconstruct it, what are the questions I would ask? And so, so I tried to write that down as best I could. And then when you look at the blog post, you'll sort of see that I say something like, look, I don't even know if this is this is how I make decisions, but it feels an awful lot like that, like what it is, And so I tried to write down a version of it and just put it out there and try to see what people thought about it. And like you said, the response was really positive. So I think a lot of people found it really helpful, and so that's how we're here today. And you guys made a really, really beautiful version of it. So thank you.
It was our pleasure. You did the hard part, which was coming up with the ideas of, getting an illustrator to work on it and make it fun was the easy part. So actually, let's take a second now to briefly look at your flow chart and sort of talk a little bit about it. So I'm going to share my screen right now and show the flow chart, and then hopefully you will be there we go, hitting share. Maybe just give a little bit of an overview of the flow chart and sort of your thinking as one sort of uses it.
Sure. So so definitely there's a really good reason why, does the experiment directly address your biological question appear right at the top. And so this is one of those things that I think, regardless of the circumstance, I think that's a good question to ask. Now, that doesn't mean that, you always do everything in a very specific way, but if you ask yourself this question, I think it's a really, really useful one. And I think I really want to make a point here that the experiments that can go into this, they don't necessarily all have to be, hypothesis-driven questions. So, when you say, what is my biological question, that doesn't necessarily mean you have to have something, a model that you think you're going to test. Sometimes this can be, for many people, it is an exploratory type question, which is sort of before you get to sort of the hypothesis-driven research. And so I sort of think that exploratory questions really fall into the sort of higher priority, more slam dunk type of experiments, because when you're looking for anything, it's a little bit more slam dunk than when you're looking for a specific thing. And so I do think that there's a lot of room for exploration, even within the framework of this flowchart. So we'll start with that sort of, does the experiment directly address your biological question? Because I think a really important thing is not just to do a experiment that can be done, but what is the experiment that should be done? So I think that, there's always questions and that you could answer just because no one has done it yet. And that may not be necessarily the thing that should be of highest priority. Maybe, there's, an infinite amount of things that no one has done yet, but not all of those things will necessarily get you towards your goal, right? Of building a story or writing a paper or, or getting a really, getting closer to your answer of the thing that that's the really burning question. So that's really, really important, I think. And that doesn't mean that there isn't room. You can sort of see in the flowchart that there are things that don't address your biological question that do still land to, yes, you should do it. And so I think that's really important, because a lot of times you don't have what you need in order to be able to do it, right? So maybe, and I think these are sometimes really, really important things too, like you have to develop tools, or you have to develop or get access to reagents or things like that you need in order to be able to answer your question. But no one's really even built those tools yet. And I think those are super, super important things to do, right? But I sort of built this thinking very much from the perspective of how to, for students to help them sort of think through what they're doing. And so while there are a ton of things that are worthwhile doing, everyone has to graduate and they have to They work within this framework of trying to advance in their careers. And so I think that is something that's really, really important to keep in mind, and something I think about a lot as an advisor, is not just, so of course you need to answer these questions, and of course you need to do the hard things and do the things that are exciting and do the things that you're curious about, and all of that is really, really important. But you, in addition to that, also have to make progress in your career. And so that's something that I thought about a lot when I was writing this flowchart. So then when we get to, if you, let's say you've got an experiment that helps you answer the question, then I ask this one that I think a lot of people have been wondering about, which is how likely is an experiment to work? And I can see how this can be a difficult question to answer, especially if you haven't done this type of experiment before. So you have no idea how likely it is to work, right? But I think what I mean by this is more conceptually, is this something that has been done before? Are there good protocols available on how to do this? Is this, or do you have access to someone who is an expert in how to do this? These are types of things that will determine whether or not it's, or how likely it is for you to be able to successfully do it, and what is your skill set? And all of these things are sort of relative to your environment, what you have access to, what's available in the literature, things like that. So this sort of likely experiment is not really about getting a specific result. It's about the likelihood of getting a result that is interpretable, right? And so I think maybe this is a point that I make in the video for the course, is that getting the interpretable experiment with the correct controls is the goal, not likelihood that some answer is arrived at, which is not what we're looking for, right? So that's what I would think about in terms of how likely an experiment is to work, is how likely can you interpret the result. And that help, I think, will help sort of navigate that fork in the road. And then we talk about sort of how long it will take. And that is definitely something that does depend upon many, many factors, right? So it could depend upon career stage. So maybe you're an expert on this thing and you've done it a million times before, and that will take you much less time than someone who's doing it for the first time, of course, right? But also it may depend on resources. So maybe there's certain ways of doing it that, you could spend a lot of money and outsource it, or do a lot, or whatever it is, and get it done in a very quick amount of time. Or you're going to have to make every buffer and make every, build everything from scratch, and these sorts of things where, it may take you a longer time to do the exact same experiment than it would in a different context, right? So those things are also really important. And then, and I think one thing that's really important about the flowchart is really thinking about having some consistent level of progress. And I think that's why I talk about slam dunk experiments and doing things right away, and whether they take a short or a long time, that these high priority experiments are things that will just keep you on that train moving towards your goal, right? So this is part for your career advancement. This is part just so you are not sucking your soul dry, So I think everyone needs some victories, right? So to be able to move towards some sort of success and have something that's always working, rather than just constantly beating your head against the wall. And I'm not saying that those things aren't important, that they shouldn't happen, but having some low level of progress is something that's just good for your sanity and also good for your career advancement. So that sort of takes care of many of the things towards the left-hand side of the chart, which I would consider high priority, and things I think about as almost things that are going on simultaneously with maybe other development of ideas and development of tools and sort of validation experiments to see how likely other things are. That if you can just have something that is progressing, and sometimes people call that low-hanging fruit. They think of that in terms of projects, but I think there's also low-hanging fruit in terms of experiments, that that you'll sort of get some sort of clarity on the biological question that you're asking in sort of a reasonable time frame, in that side of the flow chart. And then everything else is sort of referring to sort of building the framework, which I think is it's really, really important, right? And also doing things that are, that may, answer your biological question, but take longer, or it's very uncertain because you're in totally new territory, right? And you've sort of, you and no one else has ever ventured here before. And that is really exciting, right? Like that's what we should all be striving for. We should all be striving for, sort of not just to replicate something. Replication is super, super important, but it's also, I think many of us are driven by these sort of curiosity of things we don't already know. And so that's sort of novel territory, but it is hit or miss, right? And so I think that's why I was really adamant about writing into the high risk, high reward category that this is exciting territory. And it is. I mean, I think that there is, I think for many of us, there's a big temptation to spend sort of all of our time there because, it's really motivating to think of something that's sort of out there that everybody would like to know the answer to. And sometimes that is the hardest thing because people have been sort of chipping away at that for a long time, and maybe progress is slow, or there's reasons that there are barriers to progress there, or there are tools that are not yet available. So a lot of those things, I do encourage people to revisit those things every so often. So we definitely in our lab have had a lot of luck revisiting things that the tools didn't exist before, but they exist now. And so something that may have been high risk in the past becomes lower risk. As tools get developed and become more prevalent. And so that's something to also keep an eye on is to keep sort of stay aware of what's available. Yeah, looks like you're about to say something.
Thank you. No, that was an amazing introduction. And I'm just going to stop sharing the screen right now so that, and remind everyone that there are links in the chat that lead to your blog where you can see the original blog post and the newer versions of the flowchart if you'd like to pull them up yourself while we're talking, but I'm gonna stop sharing partially because we have questions from the audience, and so I'll get to that, and I just want to encourage everyone else to please keep adding questions. So this is something that came up in our course forum and is nicely stated here in our question box, which is, how do you prioritize experiments that you know are doable, but you don't personally have the skills to do yet? I think this is a very common issue, especially for maybe trainees who are just starting out.
Okay, so that's a really, really great question. And I think this is sort of one of the great things about grad school, right? This is a really intense time of learning and learning skills and sort of learning how to develop these ideas and things like that. And so I think especially when they're doable and you don't have the skill set, I think it is really high priority, in my opinion, to sort of work towards learning how to do those skills, right? So that may be something that, I think you can imagine, as the day you set foot in a lab, there could be nothing that's very quickly doable, right? There may not be an experiment that you could do quickly because you've never worked in that organism or you've never done that type of science before. And there's sort of this big hurdle at the very beginning to learning anything. So at that stage, when sort of everything has that barrier, I think it makes a lot of sense to prioritize learning those skills, right? And to see if you can get a good range of ideas on how to do that. And I really, I think, encourage people to really go out there and look what's available. So I think a tendency is to sort of be under someone's wing and do something the way someone else in the lab is doing it. And, especially as a PI who, some of the things that I know how to do that I could teach are extremely out of date, And so I think it's, learning from someone else is fantastic because you can avoid mistakes that they've made and things like that. But a lot of times, the older way to do things is not cutting edge, is not the easiest, is not the fastest, doesn't take advantage of all of the tools that are available. And so I really think it's good to prioritize, learning those things and have that be a steady progress. And if you do know how to do other things quickly, and then that can be your slam dunk, and the learning thing can be the thing that's the ongoing background type of experiment. And then doing that, which may require doing it over and over and over again, because sometimes that's what it takes to get something to get a new technique that you're not familiar with to work. But just doing that in a way that I think incorporating sort of the latest and greatest technologies along with the sort of wisdom that sort of exists in the lab.
Thank you. That's super helpful. Another question has come up on the Q&A, which I think is really sort of leading on the sort of more exploratory, difficult side, which is how much time is optimal to spend on building new tools, right? So how much, when you're heading into this, how do you know when, like, how much is a reasonable amount or not, and when do you have to make decisions about, is this taking too long or not?
So I think it's unsatisfying, but part of that my answer is that it depends. So if you need to develop a tool that it's doable to develop, but you need to tweak it or something like that, and you are absolutely going to need that tool, and we've done this before, that this is something that we know we're going to need, and that it opens up a huge amount of doors to be able to do it, and it's sort of not in many ways optional for us to develop that tool, right? And so in that sense, it must be done. But the short answer is not all of it, right? So just, you can't spend all of your time to develop tools which may not be developed. But I think when you think about it in terms of, I still need to graduate and I still need to move on with my career, and if in the worst case scenario, what happens if the worst case scenario, you spend all your time developing a tool that doesn't get developed, or it does get developed but maybe it takes you 5 years to develop it. I think this is something, at least from a student perspective, that I think is difficult, because you still need to compete for whatever job you want next. And so I think keeping an eye on the prize for what is it that you do want, and what do you have to do in order to be competitive for that. So if you are going to go on to something where developing that tool is going to be a really big feather in your cap for the job that you want next, then you could see that would be more, that you may want to put more time into that than someone who is not necessarily, it's not weighted as heavily, but their publications or whatever is more important, or teaching is more important, or whatever it is more important for so I think that's something I think of as an advisor, is what does this person want to do, and how should they allocate their time to make themselves most competitive for those jobs that they want. And so I think those type of decisions, even though they are not directly experimentally related, may impact how you decide to spend your time, or how you decide to allocate your time. And so I would say again that, it's always good to do more than one thing at a time. And I think, like I said, having that baseline of maybe trying a few things, I think, many of us tend to be able to, really, really, really focus on one thing, and really try to make a big push to make that thing work. And so, but I think we have full days at our disposal. So if you have a full day at your disposal, even if you spent part of every day developing that tool, and part of every day doing some other thing that makes some other progress, or several days a week doing one versus several days a week doing other, as long as that that one thing is really focused on heading towards your career goals, and the other thing is and those things may not be mutually exclusive, right? And then doing the thing that is progressing yourself forward, doing the really difficult things. And difficult things are so important. So, I would never minimize the difficult things, but I think everyone knows, if you spend all your time doing that, it could end in disaster, right?
Absolutely. Well, we have a couple of great follow-up questions from the audience. So one of them is, if my PI can't or shouldn't teach me, what are other ways that I can learn in addition to my lab mates? So around new skills or new techniques, what suggestions do you have?
So first, to be clear, I'm not saying your PI shouldn't teach you. So it could be, your PI may be the world's leader in the thing that you're trying to do. So they are an absolute essential resource, right? But that doesn't mean that they're the most up to date. And all I'm saying is to not assume that, if I just learn it this way from the people who are telling me how to do it, that's going to be the most optimal way. So I guess I would say, don't discount those people, but just know that those are not your only resources. So then the other question is, what are those resources, which is what you just asked. And I would say, thank goodness for the internet. I mean, I just think it's amazing that, you could, so obviously Google is your best friend, and you will be amazed. I mean, I think with YouTube and, even this, there are all sorts of experimental videos that you can watch. There's a really great, there's a Journal of Visualized Experiments that in our model organism, someone has made a video on how to do tetrad dissections. So this is just something, it's a very, a sort of technically difficult thing that even in the lab, it's hard for me to show people to do, because we don't have a camera set up on the microscope. And so for this video, they actually have set up a camera on the microscope. So to show someone on that video is infinitely better than me just talking about it while they watch me looking into the scope, right? Which is completely useless. So even if I can tell them the same thing that can be shown there, there are some really amazing, rich multimedia resources that are available online, and even just making use of your community. So it doesn't have to just be your PI and your lab, but what about other grad students that you know? And these, we have, other communities that all of us have built for, and that my students have built and that I've built for staying connected with other students and things like that. And just, making use of, people with other expertise who may be more on the cutting edge of those type of experiments. So you can still ask people, but ask different people, or find out from them how they learned it and what their resources are. So, but right now, I would say if I need to learn how to do something, I'm looking on the internet and trying to find, looking through the literature, looking for videos, looking for who might be the person who knows, So it's sort of a multi-pronged attack for trying to find the answer to that question.
That makes a lot of sense.
Yeah.
So here's another mentoring question. This actually came from our forum for the course, but obviously related to this theme is, when you're assessing how much time an experiment will take, one of the issues that was brought up by one of our course students was this issue of short-term and long-term being relative concepts. So depending on who you are, maybe you're the grad student or maybe you're the PI, you might have very different definitions for what that time frame, what short-term and time and long-term actually looks like. So how do you actually suggest people navigate that issue with their PIs?
Yeah, so I think it's really important that, so even within themselves, so them as a first year grad student versus them as a fifth year grad student are going to also have very different opinions about how long things take, right? And so I think, expectations are really, it's good to have those discussions with your PI about, and everyone knows that new people who are new, they're going to take longer to do things. It's part of the learning process. It's absolutely necessary. So to not invest that time to learn those things is a big mistake, because eventually you need to know them, right? And so it is absolutely a relative term, but I think within one person you can say this would, and if you say, oh, well, I don't know how long it would take me to do something because I've never done it before or something, that's what these, I think there is no substitute for trying to do something, right? So if you're not sure, all it takes is, sort of one or two times of trying to do a type of experiment to sort of see what you're up against, right? And that can help you say, oh, wait a second, I hadn't thought about these other factors, and if I really want to do this right, this is going to take me this long. So you may not know right away, and your PI may not know. Sometimes I think a lot of times there is a little bit of a disconnect with PI saying, "Oh, this should only take whatever amount of time," and people say, "Are you crazy? Like, come back and do it and show me this will only take so much." I think you sort of lose track of time and figure out how long things take. And so I think it's fine to say, this is how long it took me. This is what just try and explain and have a conversation and try and decide, okay, I estimate on the basis of how long it's taking me to do it, that this is how long it will take, and so just having those discussions about expectations, and, your PI will know that it takes time to learn to do things, right?
Yeah. That sounds good. But there's a follow-up.
Yeah.
From our audience, which I think is also a common issue that sometimes comes up. What do you do when you and your PI have priorities that disagree, that aren't quite aligned?
Okay, well, so this could be for a number of reasons, right? And so I think one thing is to have, actually, so what we do on purpose is every 6 months, I have a conversation with the people in my lab to find out what their priorities are, and to have that discussion so that I don't think that they have a certain priority and then they in their mind have changed their minds or have a different priority. It's really important to be on the same page, right? So I think communication is absolutely key. It could be, and I think a lot of this happens when there's sort of miscommunication on what kinds of careers people want. And so there are certain things that from the PI perspective, I would say, sometimes you are funded on a grant, right? And that grant, you have been given taxpayer money to do the things that you said you would do, right? And the bulk of that money is going towards paying people in the lab. And so, and then every year you have to report on what have I done, to deserve, to continue to have the privilege of having this money to do this work, right? And so I think sometimes that gets lost a little bit in the mix, is that there's a lot of pressure to produce something. You can look at it however you like, whether it's, but in many ways, I think we do have a responsibility to do the right thing with the money that we've been given, to spend it on the things that we've been given money to spend it on. And that is usually to try and progress on a specific type of work. And so I think that those things, and definitely people have priorities. They may say, well, look, I need to get experience doing this and this and this, and that maybe their priority isn't to, drop absolutely everything and push this paper out, right? Or whatever it is. I mean, I can see infinite number of different clashes that could happen there, right? But I think all I can say is that I think communication is key, and trying to work out a solution that both people feel is reasonable.
That makes a lot of sense. Going back to one of the sort of themes that you brought up earlier in this conversation, sort of about doing a number of different things at the same time, we were sort of focused on sort of maybe having different experiments going. But one of our Q&A watchers is asking about, now I'm a starting PhD student, what would you recommend a student do around writing? Should they be writing as soon as possible, as often as possible on the side, while also learning and optimizing experiments? Like, how do you manage sort of both the experimental part and the other parts of what it means to be a grad student, in this case, specifically around writing and thinking about publications, that kind of thing?
So you've asked me a question that I feel like very, very strongly about. And it's that you can't write too much or too often. I just think it's just so, I think it's such an important, it's not just a skill, and I don't think of it as secondary to experimental work. I actually think of it as a really, really good way to make sure you know that you're on the right experimental path. So one of the reasons, I should have said this at the beginning, one of the reasons I made this flowchart and wrote this blog post is so that I could think through it by writing it down, right? And so I think sometimes when you're trying to make an argument on paper, or to make a decision on paper, to me, I think writing is instrumental. And I think, I think we all recognize how important reading is. And I think reading is one of the top things that's important for writing. So in order to write well, you need to read a lot, and so I think all of those things that it can feel like reading is different than writing is different than experiments. And I just think all of those things are so synergistic. When you read a lot, your experiments will get better. When you write a lot, your experiments will get better. Those things are not mutually exclusive. Now, of course, if you sit at home and read and write all day and never pick up a pair of gloves, your experiments are not going to go very far, right? So it's obvious that there needs to be balance. But I think that as you spend time sort of building out all of these things and making yourself a well-rounded scientist, all of those things will progress, and they will not progress in an additive way. It will be synergistic and produce something that is greater than its parts, is what I feel, yeah.
So you've led straight into a question that actually came up, or question and issues that came up actually on our forum, which is, everyone knows how important reading is, but it also seems incredibly challenging. Like, how do you do it? How do you stay up on the literature? So how do you or your lab members actually manage to make the space to do the reading that helps support the experiment and sort of keep that productive cycle going?
I love this, and I love this even more because I know my students are watching, and I think they might tell you how obnoxious it is that I sort of, they're sort of guinea pigs for every single thing I can think of to get everyone to read more. And so we've tried a lot of different things, which, and it's just because I know it is so tempting to say, I'm going to do this experiment, and, being mired in the literature is not always the most like appealing thing to do. But we also always do, so we've tried different kinds of journal clubs where I can run down a bunch of different options there are, but I think the most important key thing to do is to just recognize that making time for this is important, right? And so even if it's doing a little bit each day, and the more you read, the easier it gets. So, I think that's one thing that you find, that people who do end up reading a lot, they get quicker at it. Sometimes, I think you'll see there, you can go through an enormous amount of literature as you get more comfortable with it, as you get more comfortable with the terms. And I know it can be extremely difficult when you're starting out, and every sentence is a battle, right? So try and understand what's happening, and every figure is confusing, and all of those things, and it can take an extraordinary amount of time. But I think that cognitive work to try and work through that and get better at it, to eventually get to the place where you could open a paper and skim the figures and say, okay, I get what this is. And then you can say, oh, I have these questions, and then only go and target for that information. And that you can become much more efficient at it in that way. But just making time for it, and making time to talk to people about it. I think that's really, really important, because you're not just trying to absorb information and just accept it for what it says on the page. You really want to read it critically. And that's why I think journal clubs and having discussions around papers is just super important, and beyond sort of summarizing what a paper says, but just really trying to figure out whether the results sort of are supportive of the conclusions, and whether the methods are appropriate, and things like that. So just if you have to choose between a way in which to read, trying to absorb and memorize everything that's there is totally, I think, not possible or worth it. It's sort of, just learning how to read and trying to look at interesting results, and you can always go back, and you're never going to have to, you can always go back and look at the paper again if you forgot some detail, right? So the point is not to try and remember every single thing about a paper. It's really just to try and read it, and read it critically.
That makes sense. Leading from reading papers to publishing papers, but also weaving in this question of what experiments to do. One of our watchers has asked the question, thoughts on publishing, should it be something small in a shorter time frame versus waiting to answer more questions to target a higher impact publication? So, those are sort of slightly different areas on the flow chart. And your strategy might be different. So what are your thoughts on that?
Yeah, so I think that's a really, really great question, and definitely one you should talk to your advisor about, because I think advisors are all over the spectrum on this question. So I don't want to give the impression that there is an answer to this question, and that, this is some sort of authoritative answer. Now, I will tell you that I think that, deciding, I think picking those biological questions is what's really, really important. And then any paper can be a big or small paper. Any paper, right? Any paper can be a small packaged thing that you just get out there to say the first, make the first result known, or you can really expand it and look at it from multiple different sides. And that line is very, very fluid, right? And that can also depend on what it is that you're looking for, right? Or what your specific goal is. Sometimes if you're saying, look, I would just like to get one more paper out, because I think it would make me competitive for X, and then that answer might be very different from, I'm really trying to, either answer this question, or I really need to, or I want to get this job with this person who is really influential in this field, that I want to really make a big contribution or something like that. And I have a lot of time or I have a lot of resources or whatever it is. And those, just again, these considerations will come into play. But that, from my own lab, I will say that I think in many ways, in terms of having sort of, I think, in my opinion, publishing consistently is much, much more important than whatever. I don't feel very strongly about high impact papers. Now, people, 100% will guarantee that there will be people who disagree with me. But in my opinion, doing good science and publishing consistently is almost always career-wise a really, really good move. And so I think many cases from my lab, I think in terms of publishing consistently and having, maybe an early paper that's very doable and reasonable, and then having something maybe that's a little bit harder, longer term, might be more challenging to develop tools for it or things like that, that having a mix of these two things is good. So that if the big thing doesn't work out, no one wants the situation where you put all your eggs into the big basket and you think you're going to aim for this high impact publication. Guess what? It gets rejected. Now where are you, right? Now you have what you thought was a really big deal and you spent all your time doing it, and you have nothing else to show for all of your efforts, right? And that this will maybe then just, whatever happens to that paper, hopefully you most certainly will still find another home for it. But just to put all your eggs in one basket, hoping that a very specific outcome will happen, it may not happen. So I think it's really, really important to diversify, hedge your bets, make some steady progress. Consistent publishing, in my opinion, is the key towards the more broader range of success.
Excellent advice. Another question that has come up on our Q&A is how to use the flowchart. So when should one actually revisit the flowchart and check the experiments again? And sort of what's the process for using this and reevaluating experiments as you go?
Yeah, so I think I made this very much thinking that we in general sort of have this idea that when you work with students to teach them how to do something, that much of the teaching happens by osmosis, that they just, by virtue of being in your general orbit, they will just learn these things, and I think that this flowchart was an attempt to say, no, these are things that can be taught, and these are things that can be used. Now, that doesn't mean that for every experiment you go through the flowchart and make a decision on how you're going to prioritize things. That if you feel, my goal was, if you feel hopelessly lost, like there's a sea of experiments, what is the method to this madness, right? And so to help you organize your thoughts in a way that could sort of help you prioritize sort of where things are going. And now all the time, even with the experiments that you've already decided to do on the flowchart, it could diverge into different territory. It may be, you may stumble on a hurdle that you realize, oh my goodness, I didn't realize I needed a long-term tool development for this. This seemed like a slam dunk, but it turned into something that's going to be much more long-term, which happens all the time, right? Science is always harder than we think it is, right? Or, it could be that you have, something that stimulates a new experiment. So where does it fit into your current framework, right? And so I think as long as, I think the right answer is to revisit it when you feel lost. That it doesn't mean that everything needs to follow this, and it's not a hard and fast rule. Like I said, many points are relative, but if you feel sort of, that you don't have a rationale for why to do something, it may help you organize your thinking a little bit. Right. And so beyond that, I think as you, and there is something to be said about experiments you're excited about. Right. And I think just thinking about in the back of your mind, is that exciting thing answering your biological question is, just reminding yourself to ask yourself these questions. So even if you don't know exactly where you would put it in the, priority list, it may still be very, very helpful to just keep in the back of your mind these questions about how feasible is it? Is it answering your biological question? And that may help you think, what, maybe I'm not as excited about that as I thought. Or you know what, this is going to be really cool. This is going to be really cool if it works, and it just might work. So I think, just have, I think more important than the actual decision tree is just the questions. And when you are really trying to reevaluate where things stand for new experiments or a sort of repurposed experiment, to just remember to ask yourself those questions to just organize your thinking a little.
Thanks. That's really helpful. So we have a question actually from someone who is starting a new faculty position and also trying to finish up their postdoc, something I'm sure you also probably experienced, and, starting new projects and new collaborations. So their question is there any aspect of the process of prioritizing experiments that are fundamentally different as you progress from grad student, postdoc to faculty? And this is someone who's really both finishing that and thinking about becoming the mentor and doing their own work.
Yeah, it's 100% different. I think nothing about it is the same. I just think that because you go through everything that we've talked about so far is when you are the person doing the experiments, right? You know your abilities, you know your skills, you know everything with respect to yourself and your experiences and what you know from the literature and all of those things. Now, when you are starting your lab, you are now having other people into the mix, which are their own places on every spectrum, right? On every level of experience or in every level of, everything. And then you have also resources to worry about. I think that's for the first time you're maybe way more worried about resources. And you're way more worried about, there may be specific goals you're looking for. Like you may be looking to get your first paper out so that you can get your first grant, so that you can, continue to keep everybody in the lab employed beyond when your startup runs out, right? So there's, all these different considerations that are in addition to the things that we've talked about so far. And I think that may even be something that comes up that we talked about that clash between what students are looking for and what maybe their advisors are looking for. And I think sometimes that can come from there needing to be many more things that need to come into the mix for making those decisions. And so, I think that I just try to be transparent with those types of things and say, look, I try as much as I can not to divert people from the things that they're doing because it's extremely frustrating to do that. But also say, oh, you know what? Sometimes you do have to, as happened with one of my students recently, she got the reviews back for her paper and we had a fixed amount of time to do the experiments for that paper before we had to return the revision. So sometimes that has to take, top priority over everything else. It's got a deadline, and so there are certain things that come up and say, look, absolutely XYZ is important, or, you may have your qualifying exams coming up. So you really need to focus on that instead of doing these other things that for which there may be, so it's just every person has their own needs and they have their own, places in their career. And you have to sort of balance their full research program with all of those considerations and those varying levels of experience and expertise and things like that. So it's a lot of different considerations that go into that. So I think that is what is really, I would say, and what most new faculty would say is the very toughest thing about being a new faculty member is trying to sort of handle the people, just it not being just you anymore. You're not the only one making decisions for yourself. You're, looking out for everyone's career and looking out for, your own career so that everyone else can have a career, So I think there is a big pressure just to make sure that the lab doors stay open, and that everyone can continue to do the thing that they love, right?
Good point. So that actually leads directly into a conversation that was occurring on the course forum, which was around sort of resources, both financial and otherwise. And the conversation was sort of about so resources aren't explicitly mentioned in the flow chart, but often that is actually a deciding factor or part of the decision-making process. So where do resources and sort of those considerations come in when you're trying to make these decisions around experiments?
Yeah, so generally I would say that I really hope that students and postdocs don't have to think much about resources, right? So obviously they do when they are being restricted in what resources they have, and then it will come up a lot. But as far as sort of deciding, I think the right thing to do is always decide what is the experiment you want to do, and then work with your advisor to find out how can we get this done and do we have the resources for it. But sort of when you're deciding in your mind what is the right, I think that's why qualifying exams and doing your prelims and everything is so important, because it's more of a thought exercise rather than I'm actually going to do everything that's here, because it should be sort of an open thought about what is the right thing to do, and then we will figure out and you need some feasibility to say, oh, I'm going to use this technology that only one person on Earth has. There's some, equipment or something that of which, two exist on Earth, that's not very reasonable, right? So you need to have, feasibility in mind, which is different, I think, than necessarily making resources a part of every decision. And I think it's sort of important, arguably, that resources are not part of explicitly every decision that you make, but the whole framework may be, like if there's, a fixed amount of resources available, that may, for example, impact what model systems you use, or what, because obviously, mice are more expensive than the algae we use, so we definitely can do a lot of things with our model system that I think would be more difficult to do in more expensive situations, right? So it's a consideration in the sense that, in the bottom line, but I think hopefully your advisors, that's what they're thinking about a lot, and that may have something to do with it. So if you can't quite, that may be another source of the clash. But again, I think just talking about it and saying, look, this is what we have available to us. This is, given that these are the resources we have available, this is the best possible way forward. But I think it's really important to just think sort of a little bit in isolation about what is the right question to be asking and what is the right way to tackle that question. And then to say, what's the best we can do, given that everybody has constraints. They may be very tight constraints or not very tight constraints, but everyone has some constraints. We don't have an unlimited sum of funds and people and hands and all of that, right? So there is always some limitations. The question is, within that framework, but you have thought somehow abstractly about what is the right thing to do scientifically.
That sounds really good. So we're nearing the end of our time, so I wanted to ask a last fun question that has come from the Q&A audience, which is, do you have a favorite story? And this could either be like a favorite good story or disaster story from your journey so far with a slam dunk, high priority, high risk, high reward, or trash experiment that you want to share.
Oh, that's interesting. So I guess I would say, there are a lot of great stories. So I think my lab would say that I get excited about a lot of cool results. We had a lab meeting today where I got ridiculously excited about something they claim I already knew and had already told me. So that could be, there's a lot of exciting things out there that, it doesn't take a whole lot to get me like very excited about something. But I do think that a lot of times the things that I find really interesting are the more exploratory things. So I think because of, rightfully, we have to work within these constraints and write grants and things like that. And there's a limited amount of funds to be given out. So, I think those granting agencies do in many cases want you to do the slam dunk experiments that they know that this money is going to something that is going to work, right? So rightfully there is some concern about that, and I do think that will in some cases, that may limit how much really big ideas can get out there. But there's also funding for those kinds of things. But in many ways, I think the really exciting things are the exploratory things where you sort of do a screen or you do a proteomics experiment and then some. We had a situation in which we did a totally unbiased proteomics experiment, and we work with a handful of genes in the lab most closely, and doing this sort of totally unbiased experiment, like one of our favorite genes came up in this proteome experiment. It was like just mind blowing. Like I'm still trying to figure out how, in what way was this like not a totally unbiased experiment that we somehow landed here. But it was just like, I still like cannot get my head around how this is even possible, with this like infinite number of, possibilities here, and trying to do what we thought was the most unbiased experiment, sort of landing right back in our backyard, So I think there's just a lot of, science is awesome. So I think there's just a lot, there's too much coolness.
That is a very fun place to end. Did you have any last final comments that you wanted to make before we end?
No, I guess I just want to say that, I think it's great, everyone who signed up for this and signed up for the course, and just sort of taking this into your own hands is just awesome. So I applaud everyone who's just participating in this course in general, and to just just keep a dialogue. You don't have to do this by yourself. Stay in touch with your fellow, grad students through the grad student Slack, which my students started, and through all the other ways in which you have to stay connected with, your peers and, postdocs that come through the lab, your advisors, other advisors for mentoring committees. There are no easy answers. Every, advisor will have a different answer to everything, but just having that spectrum of information and asking multiple people what they think, and then sort of synthesizing what you think is the best answer on the basis of all of the advice you've gotten, not just from one person, and I think that just will put you in a really, really great position of strength to just make sure that you have a community and a mentorship committee, and just to support you in your growth. And I think that's just super, super important.
Thank you so much, Prachee. This was really fantastic, and thanks to everyone for attending the Q&A. We have recorded this video, and we will be posting it on our YouTube channel, iBiology Career and Professional Development. And if you're taking the Let's Experiment course, it will be also posted in the course platform. And I think we're just about to throw some links up in the channel, in the chat channel. And there was a comment on chat about getting the flowchart in high res or in lighter colors so you could print it. Both of those are actually available on Prachee's blog post. So she's updated. If you look down below the original blog post, she's linked both of our updated versions, both the black background and the white background. So you should be able to find them there. If you'd like to know more about iBiology and upcoming events, you can follow us on Facebook, Twitter, and YouTube, or sign up at our newsletter on our webpage at iBiology.org. Which just, let me also give a plug. Prachee is really active on Twitter, so if you are interested in hearing more from her, I would really recommend you go find her on Twitter. If you're interested in specifically the Let's Experiment course or our courses on courses.ibiology.org, we actually will be releasing it as a self-paced course in a couple of months, and you can actually already register for it. So that information will also be showing up in the chat box. And we really just thank you, Prachee, for joining us today, and thank you for everyone watching and giving us your great questions. We really appreciate it. Until next time, thanks so much.
Thank you so much. It was awesome.